Hello. Thanks everybody for coming. We might have like 1 minute by the clock left, but let's get started because I'm glad to see so many people excited about data reproducibility. I wasn't sure who would show up to this session, but it should be fun. And I do want it to be very interactive, so please do interrupt any of us at any time. Is there an audience microphone in the back? I think you could go to that one by the laptop over there or just yell. We're a pretty small group. So very briefly, thank you for coming to this session. I'm Drew Jones. I'm an assistant professor at NYU Langone. And this is a subject that I think doesn't get talked about enough, but it's near and dear to our hearts, so to speak. So hopefully that'll lead to some interesting discussion. I'd like to introduce the other two panelists who will also be speaking. Yik, can you go first?
Yeah. Does this work? Hi, my name is Yik Siu. So I'm the senior bioinformatics programmer in Drew's laboratory. We are part of like a metabolomics core at NYU Langone Health.
Hi, I'm Scott Givan, and I'm the director of the Bioinformatics and Biostatistics core at the Van Andel Institute.
Excellent. Thank you, guys. Really looking forward to the presentations and discussion. But before we jump into those topics, we do have, we're very fortunate to have a sponsor for this session. This is Sean. Thank you.
Hi, good morning. So I'm Sean Martin Simpson. I'm the Bioinformatics FAS for Canada. But I be here because before working for Nanopore, I used to belong to a core. And as a bioinformatician belonging to a core, I know what it's like trying to reproduce data and when not using tools that allow you to perform this task. So I just wanted to give a brief introduction on how Nanopore is going about making this task easier for cores and for the simple scientists. So as you can imagine, learning bioinformatics for any biologist or anyone unexperienced with programming can entail a pretty steep learning curve. And it can be time-consuming even for the most experienced bioinformatician. And finding the right tools, algorithms for each specific project or approaches can be pretty difficult. So at Oxford Nanopore, we're now trying to put together time-efficient solutions, providing pre-configured workflows and guidance in finding these best tools. So our goal is to enable the analysis of anything by anyone, by anywhere, but that also means, also applies to the data analysis. So we provide tools including user-friendly end-to-end workflows to perform any kind of analysis from QC to reporting, tools to match all levels of expertise, from non-scientists to experienced bioinformaticians like the panel. Tools which you choose where to run your analysis depending on your needs and your infrastructure. So it can be on a cloud, it could be on your desktop, it could be on your laptop, it could be on an HPC, whatever. So here you can see a list of software and which stages of the analysis they cover, and what level of expertise is required of each kind and which we support. It is important to know that apart from the software controlling the sequencer, we have other software tools providing the full workflows from the specific genomic analysis. You can choose a user-friendly interface to run them on, or you can do it by command line. So as I said, if you're comfortable working in command line, but you're new to Oxford Nanopore, we have our GitHubs. We have where all our pre-configured workflows are. And we also have a pretty user-friendly, intuitive platform called Epitome. So those are our two main focus. And Epitome offers a pretty user-friendly platform. So it's click a button, select your workflow, select what your input files are, the type of output files you want, the basic standard. You can get an HTML report. You can get your VCS or your variants, your BAM file from your alignments, etc. And we provide a range of workflows. So they range from microbial analysis to human genome to biopharma to single cell. An example is our metagenomics classification, which can also be operated in real time. So as you sequence, you're getting that report generated. And from assembly of small genomes, validation of clones, your single cell, your transcriptomes, workflows, etc. We offer all these. And the epitome, as you can see here, is supported on any desktop application. So it could be Ubuntu, Windows, Mac. We also offer a cloud base where you download the software onto your device, but the analysis actually isn't run on your device. It's uploaded to our cloud server, and you can run it free of cost. And it also provides that intuitive reports, which you can then provide to customers or to other people you're collaborating with. So our epitome workflows can also be integrated into additional tertiary analysis. So if, let's say, you're used to using NF Core, because similar to NF Core, epitome is built on Nextflow, you can take your NF Core pipeline and just integrate it straight into epitome software as well. And you can optionally integrate your own pipelines if you develop on Nextflow. So we currently have established 4 tertiary analysis partnerships, which integrate into epitome. And they focus on the variants and rare results from diseases, offering cloud and supporting different kinds of approaches and methods. So we have the GenYX, Vardsum, Fabric, and Sequent. So if you want to learn more about this pipeline, our workflows, anything to do with Nanopore, please come and visit our booth, or you can come and ask me questions. Thank you.
Any questions for our guest speaker? Excellent. Okay, thank you. Hopefully everybody can hear me okay. I'm really pleased to see the turnout for data reproducibility. I think this is one of those things where you know it when you see it, but what is it is very complicated, and hopefully we're going to talk about some of that today. But again, I do want this to be a conversation, so please interrupt. I think there's a couple microphones you can go to. So first, I just want to thank my laboratory personnel and the funding that makes our type of work possible. I'm actually not really going to show any of our work today, but more about a meta discussion of how we do our work and how we ensure that it's reproducible. I think in core facilities, we have a unique opportunity to really push this direction forward. Because we work closely with investigators, we're processing data on their behalf often. So just quick show of hands, who runs a core facility, or just like this? Yes, so a lot of directors. Any staff of core facilities? Oh, excellent, an equal mix, fantastic. I think that'll be interesting if people in labs feel differently about how reproducible their work is than the people who are running the labs. But let's see, do we have any genomics people in the room? Proteomics or metabolomics? Fantastic. Like microscopy, anybody doing like instruments fleets? Okay, so these are really different use cases, right? I imagine who's transferring raw data to clients, raw data? Yes, fantastic. And who's doing analysis on people's behalf, on a client's behalf, where you do data processing? Basically everybody, fantastic. But these challenges are very different, whether you're providing an instrument fleet or you're analyzing data on people's behalf. We have a lot of custodianship of scientific work on behalf of our clients and collaborators. And we get to see so many projects, right? I know I personally have learned a lot, much more than in my PhD and postdoc, seeing so many projects across the core. So I think this audience is going to have really interesting insight into what reproducibility is. But why do we even care about reproducibility? Some interesting citations. This paper on the left, actually, the title doesn't quite betray what it's about, but trust in science is at an all-time low right now, and that's in part, there's many factors, that's in part because of high-profile situations where data is not reproducible. I think coming from the psychology crisis, where we had the so-called scientific reproducibility crisis, every sub-discipline is also facing this type of crisis. And there's debate about whether it really is or isn't. You know, what do we mean that some study didn't pan out? It's pretty complex, but hopefully you can see that this is a growing topic of interest that we, especially in cores, I think can help address. I don't know why this is kind of glitching out like that, but and related to this thing of reproducibility, we have new trends like self-citations and paper mills. So I think reproducibility, depending on how we tackle it, can help tackle some of these difficult problems that are undermining trust in science. And that's why it's such an important topic to us. And then we also have high-profile things. Does anybody know this icon? Fantastic. This is Retraction Watch. If you've read this blog, and many, not all by any means, but many of the things you'll read on Retraction Watch come from sloppy data processing, irreproducible data processing, honest mistakes, and also not honest mistakes. So I used ChatGPT to make this graphic last night, and I love it because it's a mess. You can see all the hallucinations. Like, it has this idea that you know, the data is at the bottom of this conceptual pyramid with biological mechanisms and reproducible science at the top. But it's a fuzzy picture. What is data reproducibility, and what is the science that's based on it? We have many different levels, which is one of the main things I want to highlight today. You know, is some biochemical mechanism actually true across multiple models? Is a disease state like Alzheimer's, are mouse models of Alzheimer's reproducible? You know, that's now a hot question. Does reproducibility mean that if you do the same experiment in the same mouse with the same disease and the same treatment, that you'll like get supporting results, like the same type of results? That's one sense. Or does it mean that specific set of samples, maybe that 20 samples, do those generate the same results if you rerun the samples in a new data set? Or does it mean you have a data set and you get the same p-values back, which feels like it should be easy, but has anybody, who raise your hands, who uploads data to repositories? Good, pretty much everybody. Who has downloaded data from a repository? A little bit less, but impressive, fantastic. And who has actually tried to process that data from a repo to see if you get the same result? Okay, fewer, we're losing people, but a lot, that's fantastic. Anybody brave? How did that go? I'm seeing some thumbs down, some handshakes. Yeah, and no thumbs ups? Not one person, wow, interesting. So how do we deal with these layers of reproducibility? There's been a lot of really good movements in the field. Many of these supported by ABRF. Research Resource IDs, RRIDs are a great thing. We try to put them in papers, but you know, a laboratory is dynamic over time. Who's leading the laboratory, who works in the laboratory, it's a little bit nebulous what a resource ID is, but it's a good direction, and this is something we should all embrace if you're not already using RIDs. They're a good direction. Are most people using some sort of LIMS software, iLab, or one of the, okay, fantastic, a lot of iLab users or LIMS in general. These are great, they help us track our projects, right? Who's happy with their LIMS? Ooh, wow. Wow, okay. That's interesting. Okay, but we have to have them either because we're told we have to have them, or they're helping us do something that's important, right? We also have electronic laboratory notebooks now. Who's made the switch from paper to eLab notebooks? Okay. Yeah, I was actually a huge Luddite about eLab notebooks a while back, and I'm a convert now, but there are really important trade-offs, especially with traceability. It sort of appears extremely traced. We have timestamps of who did what edit when, but unlike writing in a notebook with pen and paper where your handwriting is authentication of who's doing it, I have a lot less. I don't know. If you see a timestamp for one of your people editing Benchling, do you really trust that it's definitely that person? Is there a doubt about that? Yeah, there's interesting trade-offs, but it's searchable and scalable. I don't have time to talk today about my lab strategy. Down at the bottom, you probably can't read this. It says Helm. These are for our workflow, we call them Helm numbers. These are harvesting, extraction, liquid chromatography, and mass spec protocols. We have a huge database where we can mix and match little protocols. So I can tell a set of 4 numbers to anybody in my lab and be like, okay, it's that type of project, gotcha. That's something to think about if you don't have a database of protocols. But there's no real consensus on how to do that, and I think that's an interesting discussion. Who here uses code to process their data? Basically everybody. Who here is generating code to process data? Wow, a lot. Fantastic. And are you using GitHub to share your scripts? How often do you download software or scripts from somebody else's GitHub page to try to do something? Good, good. A little bit less, a little bit less. Excellent. So GitHub's an industry standard, not just in science, right, across all computing. And it's a great tool. So if you're not using it, I would definitely recommend that you do. And who's using some sort of workflow manager, SnakeMake, Nextflow, Galaxy, something like that? Excellent. And are you happy with your workflow managers? 4 or 5? Okay. Interesting. So we're all doing these things and we're all unhappy with them. That's I think that says a lot about the next couple of slides. And then, has anybody had to upload some sort of Docker container or CodeOcean for a publication? Journals are starting to enforce this a little bit more now, especially high-profile journals. Yeah, a couple people are using containers. Are you doing containers because you have to, or because you actually use them in your work? Yeah, you like to use containers. Okay, excellent. So I think I've run through, and we've already talked about repositories a little bit. So we're all using these tools, but we're all a little bit unhappy with how they work because we have very complicated needs and everybody's lab has different needs. And to throw a wrench into our operations, the NIH data sharing mandate is now in effect with very little support and clear definition of what we're supposed to do when it means to share all of the raw data. Have your institutions addressed the NIH data sharing mandate? Yes? No? Almost no. Yeah. This is a huge burden that theoretically we're supposed to be doing. And there's not really an answer on how we're supposed to do it. What does raw data mean? Where do you share it? Is there funds to do this? I think it's a great direction. It's a great vision. But despite all the tools, it's not really it's not really enough to share data. If you read a Nature paper and you want to see if that result is quote reproducible, what's the process? You go download it. You maybe try to download their capsule if they did that. Maybe the data is shared, maybe it's not. Maybe you have to configure your environment to run it. It's honestly extremely difficult, and that's the kind of questions I want to get into and see what everybody's struggling with and where we can go with this. So I'm gonna switch to the next panelist now, but keep this question in mind of, have we achieved data reproducibility with all of these tools?
I want to get that view you had, Drew. I'm pressing the wrong button.
It's working?
No, but I want to see my notes, like you could see your notes.
Oh.
Otherwise, I'm going to be lost. Anyway, so I'm Scott Givan, and like I said, I'm the director of the Bioinformatics and Biostatistics core at the Van Andel Institute. And I was just going to give some information about the VAI because I'm sure some of you know about it, but I would guess that some of you don't know about it, and there's some relevant points here for what we're talking about today anyway. So it was founded in 1996, and this was a slide that I asked the communications team. I asked them, what would you want me to say about the VAI? And they said this stuff, and they said, do you want us to make the slide? And I was like, yes, make the slide. So this is the slide they came up with. So it was founded in 1996, and currently there's over 500 scientists. And probably what this group is most interested in is that there's 9 cores at the VAI, and we are all actually co-located in the same place, and there's actually a very centralized management architecture there, so it's well managed, and we're all close to each other. And as a bioinformatics core, we're the only core that's not generating data, right? All we're doing is analyzing data. And so we have relationships with other cores because we're so close and we're collaborative that we play a role in some of their data generation pipelines, but as a core ourselves, we do not generate data. And that will be important. These are the people. I may say words like I and me and we in combination with words like work and effort, but it's never me, it's always these people. Actually, of my group, there's only one here right now, is Ken Lau, who's in the upper left there. What's important about this slide that I'll come back to is that I've been there for, it's going to be 6 years. And there have been times early on when there were only 4 full-time analysts. And so we've grown fairly significantly over time, and that's caused some of the priorities for us to be consistent, and I'll come back to that. I'm also going to probably refer to my core as the BBC, which I know if you're from the UK probably sounds like a really bad idea, but it's okay here in the US, I think. It doesn't raise the same issues. Okay, so I was at dinner the other night with my family before my trip, right? So the family wanted to go out to dinner, and because Dad's going on a trip, at least that's what I like to think about it. My kids were probably dreading the dinner, but anyway. So somehow my daughter keyed in that I'm going on a trip, and she's 16 years old, and she's about what you would imagine a 16-year-old is like, and so she says, so Dad, you're going on a trip, and you're going to give a talk, and people are going to listen to you? And I was like, I think so. But of course, then and there, staring at the endless salad bowl in Olive Garden, I have the imposter syndrome, like, why am I talking about this? And I don't think there's really best practices for this. I think that I'm going to go through what we do as a core, but I'm sure there's people in this room that have better ideas, different ideas, better ideas, and so I think that's what part of this discussion is. I'll talk about what we do as a core, but I'm happy to discuss and find out what other people are doing too. So also, I know that it's popular to ask ChatGPT for definitions of things, but I took the relatively novel approach of asking my head, what do I think is reproducible data analysis? And so this was the definition that I came up with. And so this is my gift to you. You're free to use it as much as you want. So there is there's some space for movement here in this definition, right? Like, I'm not saying that you're going to get the exact same results, because we all know that tools change over time, and so you may not get the exact same result anytime you go back and redo an analysis or whatever. So basically what I'm trying to say here is that if given the same set of data and the same set of steps, you're going to be able to reach similar conclusions or the same conclusions, even if it's not the exact same p-value or the exact same threshold change or something like that, right? So you're going to get, if you have the same data, you do the same sort of analysis using the same software, by name at least, let's say, you're going to come to the same conclusions. So what I'm mostly going to talk about is pretty much our internal mechanisms for dealing with this. And they do scale out to external mechanisms too, which I'll go over. So one of the, hold on, I'm reading my notes here, sorry. Oh, so as I mentioned a couple of slides ago, the BBC has grown significantly, the bioinformatics core. And so if you think about it, as you incorporate new people in your group, or as we incorporate new people into our group, new ideas came in, and you don't want to shut down those ideas because sometimes they're good ideas. So your methods can change over time, and yet we want to be able to give consistent results to people that are our clients that are coming in the door. Most of our clients, at my core at least, are internal, so they're coming from the institute. And so you don't want to have different results coming from different analysts for the same data, right? You want every analyst to be delivering roughly the same results for any given data set. And so we needed to build in mechanisms to have reproducible data within our group, right? We want to have consistent and reproducible data generated by the analysts for the given data set. And so we had to come up with mechanisms of ways to manage that. And so I'm going to go over those mechanisms, and I'm sure that from the show of hands I was getting before, you're probably all familiar with most of these mechanisms, and it's probably not a huge surprise of what we're doing here. One of the key things that might be different for our group that is from most bioinformatics groups, I would say, is we have two teams of people that were up on the slide before. There's a biostats team and a bioinformatics team. They are separate teams in my group, but we obviously interact a lot. And so I asked both groups for feedback in terms of what do they think of in terms of how we support reproducible data analysis within our group. And the stats team right away said, well, the experimental design is really important. You have to start with a good experimental design to be able to have reproducible research. And also have things like power analysis so you can predict the sample size you need so that you're going to avoid biases and you're going to have well-sampled data. That's another key to having reproducible data analysis, right? Even whether it's in our group, it's other groups at our institute, or it's outside of our group. You want to be able to be coming to results that are well represented within the data set. And then also the analysis you do, the statistical test you do, the model of the statistical test has to match the data type that you're working with, which is not always the case, we find, with published research. So that's sort of from the biostats viewpoint. From the bioinformatics, the rest of the slide is essentially bioinformatics related. So we do use a standardized compute environment, but we don't really work with containers per se directly, but I'll come back to that a little bit. Most of the bioinformatics group is doing stuff on the command line, so we have a Linux cluster, and that's where we do most of our work. My group, the bioinformatics group, manages the bioinformatics software on the cluster, right? Because there's our group, that's the core, but there's also labs, and there's certain labs that are really heavy bioinformatics labs, and they're doing the same kind of stuff. And so we tend to manage the software for the whole institute. And so we make heavy use of things called Linux environment modules, which have been around forever, but it's ways to deploy software on a Linux machine or a Linux cluster, and you can have different versions of that software, and it's a way to manage that whole environment. And so we do that as a core so that we can reproduce data from now and from previous results if we need to walk backwards. My group also heavily uses R, and instead of having everyone run that on their own laptop or their own computer, we use a centralized R Studio server. We use this on demand, which is fairly common these days for this sort of thing. So we have a centralized RStudio server that the whole group uses the same version of RStudio. We have a relatively limited number of different versions of R that people use and stuff like that. So we try to standardize that as much as possible so that we don't have every person in the group using a different version of whatever they're using. And we also use Python for scripts and things like that. We do use fairly heavily, we use workflow engines. We use SnakeMake specifically, but we find this is probably one of the most important ways for us to be able to do reproducible things, and also to pass on reproducible analyses to people in the institute, and also publishing papers and things like that will often have a workflow associated with that. And the workflows really sort of formalize the different steps of the analysis we're going to do, which again contributes to being able to do reproducible research. So rigorous quality control and the analysis and the output are all sort of standardized in terms of the workflows. And they're obviously tailored to the different types of analysis that is happening there, whether it's RNA-seq or ChIP-seq or something like that. We do use a project management software called Jira, which is not probably news to anyone here, but it pretty much is the only software we pay for. I feel pretty strongly about having freely available software when we're working for scientific research, but we do pay for Jira, and it's really been a godsend from when we were 4 people to now when we're 10 people. It really helps us to keep track of projects, and again, it's all sort of these little parts of it all sort of go into one grand strategy of being able to do reproducible things and being able to track what we did and talk about what we did in terms of, what we did. I can't remember what I did a week ago, and so, we can go back and look at something we did a year ago and figure out that we did these steps and be able to reproduce that if we need to and build on it for if there's new data coming down the pipe and things like that. We do try to obviously track software, record software versions in every report we give people. So when these workflow engines will often generate a report, or with a little bit more work downstream of the workflow engine, there'll be a report. We track all the software and the software versions, the library versions, and things like that. We also make a fair use of Shiny apps, like if there's a certain sort of analysis that we want to be able to share with other groups, and they want to do the analysis, either build on our analysis or redo our analysis. So again, reproducing our results, we will often formalize that analysis as a Shiny app so it's easier for those groups to use. So Docker containers, we don't do, as a group, as my group specifically, we don't do a lot with Docker containers. However, our whole HPC is based on Docker containers. So when we're working on a node, it's actually not a physical node, it's a Docker container that represents a node. But we have done a little bit of work with Docker containers, and if we were called on to do more work, we would, but we don't really use a lot of Docker containers at this point. And then Git, as Drew was talking about before, is a way for us to track our versions of software and be able to fix bugs if they happen and have multiple people contribute to projects. And Git was actually one of the things that sort of is a bridge to doing external reproducible data analysis. Version control software has been around for a long time, right? Like RCS and CVS has been around, and then Git is really the dominant one that's happening now. And Git also has this great thing called GitHub where you can share code. And I just wanted to make it, again, the point that we really try to focus on using open source, which when I was putting together this talk realized that open source does not actually mean free, which I don't know how it would work where you'd be paying for open source software, but really the more important thing for us, I think, as a group is that we want our analysis to be supported by free tools that anybody would be able to use. And so often they're open source, but they don't necessarily have to be open source, like they can just be free. So there's Git version tracking, which I was just talking about within our group. We use it to collaborate within the group, but it's obviously when you post things on GitHub, it's a way to collaborate within your group and externally to other groups. And it's also a way that we share code if it gets published in a paper and they need to share the workflow for that. We almost always will want to include example data so that I think a lot of us know that like example data is key for debugging something that's not working on your system. If there's example data that you know should work, it helps you debug the pipeline. And it also is going to give you results, and the example data will let you prove that the pipeline is working in your system. We also really enforce including a license with our stuff. So we use GPL2 now, which pretty much allows you to do anything, even commercial stuff, as long as you track the provenance of like you give credit to the people who generated the code. There's different licenses that you can look up on GitHub that you can publish with your work, but it's really important because most of the license will relieve you of any liability if someone wanted to sue you for something happening with that your code generated results that cost them money. Most of the licenses that you can post on GitHub are going to free you up from any liability that would happen in that situation. And I know my organization is really sensitive to that sort of thing. Again, workflow engines, as I mentioned before, is a really useful way to support external reproducible data analysis because you're giving the same workflow that you used in-house, you're going to give it to an external group, so they should be able to reproduce that. We also really try to provide extensive documentation within our workflow. SnakeMake there's going to be a config file for SnakeMake, and so within that config file, we usually have really extensive documentation about what each step, what each configuration option is going to do to the pipeline or how you can tweak it. And then finally, in publications, we try to be really specific about anything that you would need to know to reproduce the experiment or the results, right? Like even command line options in the paper, we to put those in the paper so that people know like the specific command that we ran on the command line so that they can reproduce it in their groups. And obviously posting things to GitHub is, as I just went over, is important. So really, if I think about the challenges that we are dealing with, a lot of it has to do with discipline, right? Like a lot of it, basically all these things have to do with someone walking in your door and they need the results right now and you want to get them the results right now. You often skip steps in the process. And if you don't go back and fill in the holes that you didn't do, like, you know, apply the license to your GitHub repo or add the certain documentation that you need to add to your script to make it make sense, then that's a problem. And so we deal with that on a daily basis. Also, edge cases or evolving technology, or I guess it's sort of the same thing here, is like as technology evolves, your pipelines are going to evolve, and so you need a way to track that so that if someone wants to reanalyze previous data, you can go back and perhaps use a previous version of your pipeline to deal with that. And also, if someone has old data and they want to build on it with new data, being able to know what you did before and how to build on it is important. So evolving technology. And then just shortcuts. Again, it has to do with you know, the PI coming in the door and wanting the results right now. You often will take shortcuts. Dealing with the impacts of shortcuts is important, and you need to go back and fix like what you didn't do. And often that's hard to do when you have the next project coming in the door. So those are all sort of challenges that we're dealing with respect to that. And that's my slides. Thank you.
Showing? Okay, great. So my name is Yik Siu. I'm the senior bioinformatics programmer at the Metabolomics core at NYU Langone Health. So I'm working with Drew. Sorry, can you guys hear me clearly? Good. So thank you, Drew and Scott. Like, give you like a very general, like sort of like overview, like about why and how to achieve sort of like a data reproducibility in your lab. So here I'm actually going to sort of like focus on a specific area that's called workflow sensitivity. So, I mean, this is our typical workflow for our like client core member interactions. So we are a metabolomics core, so we do LC-MS. Usually it's like we will do initial client consultation. Client will bring their sample, and our core members will process it, acquire the data, then we analyze it. We have a lot of in-house scripts for to achieve like a Python or R to process the acquired data. We generate like plots for publications for the clients, and most of the time they will be happy with a significant p-value and go on and publish their paper. So again, this is our typical workflow. Everybody's happy, always. But honestly, if you really think about it, sometimes the client will come back and try to, for example, 2 years later, trying to replicate like the same experiment with a new grad student. And sometimes you cannot replicate the result. So you try to ask the question, like, what's going on, right? So there's like various different sort of like problem can be introduced at various different sort of like steps throughout the whole workflow. First of all, from the client side, which we have less control about what's going on, but still, if there's something we can do, we can sort of like sit down with the client, try to figure out exactly what's going on, how the experiment has been done, what kind of control we want to have. But still, like sometimes the client will just come back with a bag of tubes in a plastic bag and say, "Can you process it?" So that means like that part, even though I have less control, but still like a slightly different changes by like how your client's process the sample can causing your downstream analysis to produce a different result. And also when the lab members start processing the client samples, what's the difference is causing a different result? Like, do you use different buffers? How was the instrument was performing on that specific day? What's the difference is like an instrument setup compared to, for example, the data generated 2 years ago or even 3 years ago? And the next thing is the thing I'm going to focus on is like we have a lot of like in-house sort of like script to processing the raw data we generated. So how exactly it's like how robust is your pipeline actually? Have we ever did like a systematic analysis about how it's performing with different data, different parameters? So that like all the things that add up together, there's like a various different questions we want to address before we can confidently say to our clients that this is the reason 1, 2, 3 why we cannot produce the original data set, or this is the reason why it actually works. So I'm going to focus on the specific on the pipeline side. So when you initially sort of create a pipeline, there's like a couple things you should do as a core, sort of like informaticians, because as a core, a research core, we're actually in a very unique position, as you mentioned. We're going through like a various different projects with different contexts. And most importantly, we don't really have like a lot of stake on the actual outcome of the project. So we sort of like be able to dealing with a more objective, like sort of like a stand. So there's a couple of different ways you can sort of like test the sensitivity of your workflow you generated or you created. One is local sensitivity. Like if you sort of like fine tuning to change the parameter for each like step, like a little bit, were you able to get the same result? Were you going to get a different result? Or all the client's significant p-value is gone? That's the one thing you want to like figure out what's going on. For the global sensitivity, you want to sort of like moving the parameters in groups and figure out a specific combination of parameter changes, how that is going to affect your final data you generate for the client. So this is just like an overview about like the same topic. But this is like generally the two things you want to test when you have a new pipeline being generated. So you can provide sort of like a higher level of confidence when you generate the final data for the client. So why do we even want to do it? Like usually it's like you create a pipeline, you run the data, you get a significant p-value, client's happy, you're happy, they publish paper, everybody's happy. Why do you want even test different parameters? Like it has been always working like for the past 300 projects. Why do you want to test the different parameters? The first thing is like we want to know if we slightly change the parameters, can we obtain the same result? But like, or if you get it like input like slightly different version of the original raw data, can you get a similar result? Basically, it's like if you put junks in, you should expect junks out. If you keep putting junks in, you get gold out. That's sometimes good, sometimes it's not really good. And another like important thing, the reason why you want to do it is because you want to figure out which parameter is actually important, right? Like because when you first create a pipeline, you read the paper, you figure out which parameter you want to put and control. But sometimes one or two or combination of parameters simply just doesn't affect your result a lot. And maybe you can remove those and hence save like a lot of computational time, hence resources. So this is just a very fun example about why trying to analyze your robustness or sensitivity of your workflow is important. Say we have two groups of animals. We want to figure out whether that group is dogs or cat. Say our pipeline is basically drew a red circle on the upper like right side, and with a smaller size. Then if all the, majority of the animal we circle inside is dogs, then we say the whole group is dogs. If it's all cat, then it's a group of cats. So that's our original pipeline. Everything works fine if that's how the data distribution is. However, it can give you false positive results. If you have a group of dogs just happen to like to stay over there because of apples, and the cats hate it. So you will always get a significant result, but the actual what's about the whole context is actually incorrect. So in this workflow you designed it, if you just slightly switch to the location you are looking at, you will get a completely different result. Like the previous like significant differences is actually gone. Now it's just like cat versus cat. So what about you switch to different like a parameter for different? So this is the local sensitivity thing I was talking about. Changing parameters one by one and see how it affect your pipeline. Here we change like the actual size you're looking at a little bit bigger. Again, you get a different result. Or you can change the different parameters that's supposed to be useful, but actually turns out not very useful. You change the color, right, of the circle, but doesn't really affect your final result. So that's the reason why doing this type of sort of like systematic analysis can help you figure out whether your parameters are important and how your parameters or your pipeline is by changing slightly different directions, you can might or might not get the same result. So this is like another example about global sensitivity analysis. You basically change a group of parameters together and see how robust your actual workflow is going to be. Again, fitting with the new data, how your pipeline is going to perform. Are you still getting the same data, same result? You put a trash in there, you should get trash out. If you get gold, you need to look into why that happens. So another key concept is like p-value. So personally, I envision p-value is basically one instance of or you feed the raw data through the whole pipeline. You generate a significant p-value. But here we want to actually cover is the overlooked area, is like how exactly your pipeline is actually performing. Within if you fit into different data, different parameters, like that's sort of like we're moving away to the final sort of a conclusion. We actually focus on the actual performance of the workflow itself. So this is like instead of cats and dogs, this is like our one of the small part of a typical workflow we're using our metabolomics core. So what it does for this workflow is basically we generate a lot. The first database is basically a bunch of scans generated from the LC-MS sort of like experimental workflow. The on-grid, what it does is basically simply go there to the raw data, try to extract out or determine a list of potential features that's existing in your raw data. And then the skeleton step is basically taking that list, then it goes back to the raw data set and figure out for each features how much ion, or how much like how high the intensity it is for that specific feature. So, and then we go through sort of like a blank threshold correction and imputation step to get a final table on the right side. Basically, it's a list of feature or untargeted metabolite names and the corresponding intensity and metadata you get from the raw data. So as you can see, there's roughly 9 different parameters through this very simple workflow. So what if we, all the value you see is actually our default value we use. For a very long time, this works, but like sometimes you keep like feeding different data. As I mentioned before, as a core facility, we're actually in a very unique position because we want to know exactly how your workflow actually performs if you, say, switch to different parameters and whether you can achieve the same result or not. So here is basically a more one of the example for us to like sort of like gauge how robust the workflow you just saw before with 3 steps. The sort of like a pinkish or red color shows like if you go back to the one before, what we're trying to determine is if we switch sort of like modify any of the workflows, whether we will get the same raw data, sort of like a final data back for a specific row. Do we get the same feature with the same retention time, with the same like peak high we generated from the raw data, or not? So if we do see that row at the final step, it will be present and show as a red color. If we don't see that, it's going to be a white box. As you can see, like some of the metabolites or untargeted metabolites we actually see is quite robust. No matter how you modify the parameters, it's always going to present in the final data set. While others, for example, the last one you see, it only appears when you give it a very specific or parameter combinations. So that means this data result is actually very fragile. And this data, I don't have the plot shown here, but this actually gives a very significant p-value. So again, the point here is not trying to see whether this makes sense or not, because in a different scientific context, this actually makes perfect sense. But again, we want to do this because we want to gauge how exactly your workflow or your new sort of like a build up, like a workflow actually performs. You want to sort of like have an idea about the thing you use to sort of like process your data. This is, again, is the same data set. The y-axis is basically a combination of both the number of missing data when you change a parameter and also the extra data you get, sort of the new data you get while changing the parameters. So basically the 1 right here means is the ratio of change. That means this means it's our default value that we default parameter we use to sort of like process our like a simple three-step like a workflow. By moving left, you basically make the parameter it's a little bit smaller compared to your default. For example, if your default value for a specific analysis is like 10 or 20, for the step 1, you just slightly modify into, for example, 19.9 and see whether how the final data you generate. Do you get more data, less data, different data? As you can see, like actually for different parameters, you have different response curve. Some of them, for example, the I think is the threshold LD is actually very robust. It doesn't really like affect the final result like a lot compared to either the red line, which is the mixed signal, or the green line, which is the retention time tolerance, sort of like a parameter change. So this sort of help with 2 things. One is you sort of like have a global view or a better understanding about how your workflow performs. Another one is for parameter tuning. Now you can see that there is some like a asymmetric response for specific parameters. For example, the green line you show here, which is for tuning the retention time tolerance. When you move to a smaller size, you give like smaller parameter compared to before, you get a bigger differences for every steps. While you will move to the right side, which is trying to make the number bigger, it doesn't do that much. So here on the right like plot you see is basically we're trying to obtain a more symmetrical response for the specific parameter. For this specific parameter, we're trying to change the default parameter we use for this pipeline. Sorry. So no matter which side you move, you always get a similar response. Again, like that's really depends on the actual context of your scientific pipeline. But again, obtain this sort of a global view, or like a sort of a global understanding about how your pipeline performance is very important. Again, this is just a sort of a summary about why we want to do this for three like major different reasons. Thank you, Paige. If you have any questions, please let me know.
Thank you, Scott and Yik. So I think looking back at all the approaches and tools we're talking about today here, we're having to go to this complexity because science is more complicated than it's ever been before. And we also have a lot more responsibility than we've ever had before, especially in cores, for making sure that the data we're generating for people is robust. Hopefully the workflow sensitivity analysis idea is maybe a new thing to you guys. Maybe you consider that part of optimization of your pipelines. But this is the sort of thing we're starting to think about in our collaborations in terms of, where did these optimized values come from? Are these good defaults to run on a pipeline? Should settings change based on the data or not? Those are all very difficult questions to answer. But looking back even at the very high level, like LIMS and notebooks, there's so many sources of potential irreproducibility in our science. And last year's ABRF conference had a really interesting discussion about various software for helping users track metadata, discussions on how we can improve data repositories so that they're more usable. But I think one of the themes that I've seen across all these tools is the tool helping you do your work, or is it an extra piece of work that you have to do to fulfill something? And ultimately, we're all flocking towards the tools that help us actually do our jobs better. And the tools that are just extra work that we have to do, sometimes we have to do it because we have to do it. But that's not the tools that we want to be using. So I was really fascinated when not one hand was raised about whether they're happy with their LIMS software. We have a lot of opportunity to improve in so many areas: data analysis, workflows, repos, et cetera. So I want to show the group one more future direction. So far we've been kind of talking about standard practices. Again, to Scott's point, I don't know that we can call them best practices. This kind of community is who should be figuring out what best practices are in these environments. But I want to show you a research direction where we're going. So you can drive the demo. But in our environment, and I think many of you have the exact same challenge, the data processing is so complex, but you wish you could share those capabilities with your clients. How often do people in your labs have somebody come back like a year after a data set was generated and say, hey, can you take that data set and we actually meant to look for this instead, sorry. And you're like, oh my goodness, I have to go find the data. If you save your R histories, maybe that's not too bad. Or maybe you've updated 5 versions ago since you've been adding tools or libraries, packages get updated. It's a huge headache. So what we've been working on for several years as part of a very large collaboration of about 13 laboratories is we've been developing a tool that we are trying to balance these two demands. Having that custodial responsibility that if we generate data for somebody, we can prove that it was done responsibly and reproducibly, but also we want to share that capability to analyze data. And those two motivations are often in conflict. I don't know if anybody lets users come into their laboratories and use computers to process data. I know like in Flow, that's extremely common. We have some data sets like that. People come into the lab and use our software. Can we guarantee that they're doing that in best practices? Are they applying the right parameters in the software? Do you trust if they export some image out that's the same image you would have gotten as an expert in the technology if they had asked you to do that image? I think there's a lot of questions about that direction. That's where I want to get into in the Q&A section. So you maybe briefly kind of summarize where we're going with this tool, but this is a tool that we're hoping to release hopefully later this year.
So what you're seeing here, let me zoom out a little bit more, is actually a platform for workflow management. As you can see, like, it's the same like three-step data processing pipeline I showed you guys before on my slides. Here is basically one of the like in-house pipeline we use for analyzing transchromatic data and metabolomics data. As you can see, each purple node is basically one of the Python script we write like in-house. So as you can see, you can envision each purple node is basically an instance of calculation while you're using a specific script with a combination of your input files, let me zoom in for the green part. Basically, it's like, what do you want to do with this script? What kind of input and parameters are you using? And what's the environment, calculation environment you use for this specific calculation? So we are, let me actually zoom out a little bit more. So for this like a platform, we want to achieve two goals. One is trying to make the sort of like informatician scientists to use different pipeline easily. Another one is like help the actual savvy sort of like informatician to track the work, what kind of analysis has been done, what kind of script they're using, what kind of version they're using for specific analysis. So for this tool, we actually, sorry, it's hard to do with the mouth. So if you are sort of like a regular bench scientist, you want to analyze your workflow with a specific raw data. For here, you can just simply doing either drag and drop to select like specific scripts. All the scripts you're saying is actually developed like in-house, so we don't have like it's not like pre-existing scripts, because for our lab, usually the data is like we analyze is quite novel. There's no pre-existing ones. You can either drag and drop for a specific like analysis using one single script, or you can simply drag and drop the whole workflow that has been created by our bioinformatician to regenerate the whole result. Oop, this is a bad design. Sorry, the screen is. This is the same basically workflow you see before, and you can basically track exactly what has been done for this workflow that has been when the workflow was first generated by the bioinformatician. Because the way we can do it is by tracking the provenance key, as you can see here. The provenance key is basically a combination of information you want to use to generate the final result. That means is what kind of input file you want to use, you used for this specific calculation, what kind of script you use to process the data, and for what type of like parameters you put in with this script. Here we did something like slightly different compared to Nextflow for result hashing. We actually trying to track the scientific intent of a specific calculation. That means we don't want to track the changes due to update of library, or simply you use a different like version of, for example, Linux system. Because that's not exactly when you first created created like a script, that's not exactly trying to do. Because what's exactly trying to do is you write a script, you want to process data by running a specific combination of like parameters. So we try to split out those two things. One is the scientific intent, another one is just for the environmental changes, which is not, sometimes it is like important, but sometimes, most of the time is actually irrelevant compared to the scientific like process you want to do. And the combination of these two is actually the provenance key. I'm showing you on the first row right here. So the provenance key, again, is the unique identifier for a specific calculation instance. So by doing that, we can actually avoid any new calculations if you use the same combination. Nextflow also have the very similar function. So that means if you try to regenerate the same thing that you have been run before, you don't have to run it again. You can, for here, in our case, you can simply go to the previous calculated folder and you grab the result. So this is going to be really helpful when you actually initiate like a very large computation. Again, because we are tracking by provenance key, we can actually track the state of the whole workflow because we don't really repeat the calculations. That means each calculation will only be calculated once. Hence, if we try to merge all the specific provenance key of the whole workflow, you can actually map them into a new state key. That means it's a unique combination of various different calculations at a specific time point. So we actually, using this tool, can achieve two things. One is tracking the specific individual steps. Another is track the overall sort of like a state of the whole workflow. Because we're doing that, we can actually track a very interesting thing. We call them like in lineage history. Actually, let me show you this. So every workflow is actually always start from an empty canvas. Oh, sorry.
I'm sorry if you guys already mentioned this, but I know you said it was in collaboration with a few labs. Is this something you guys are custom building, like as a software? Is this something that you could potentially use out there? Is it a tool that already exists?
No, we actually developed this tool completely by ourselves from scratch. Yeah. But we do use some like well-known libraries for like a framework for both the front end and back end though. But the whole concept of the actual development of the tool, we actually do it like in-house by ourselves. So again, just go back to the state history. As you can see, like every actual workflow comes from like an empty canvas. So we call them root state. But whenever you start initiating new calculations, you always start creating a new combination of the provenance keys. Hence, that means the more like calculation sort of like you initiate is always coming from a previous state of the workflow. By doing that, by tracking those information, we can actually plot out the whole evolution of your workflow. You can name it. For example, if this is a final publication, you can easily trace back which step you sort of like invoke it can actually is like responsible for generate the final publication data. So that essentially means you're not just showing, sorry, it's hard to move around with this trackpad. So you're not just tracking each single steps, you're actually tracking the whole workflow history. So that means when you publish a paper, you can simply publish this whole workflow history. Hence, you can basically satisfy the NIH mandate because this is the history of everything you did for this analysis, including the published version of your workflow or final data result. Is actually also involving different parameters you tried for this specific analysis. So it's literally everything you did in a history sense. So we're pushing that because of transparencies, because we don't want like people just handpick the single results actually working. We want to see how you actually analyze the data. Is there anything you tried, or is there anything you haven't tried? And by copying and pasting this whole workflow, you can actually extend the analysis with different parameters when you actually, for example, you see a Nature paper with this specific history, you can copy and paste back to your local instance, and you can run it with new parameter and see whether you can get the same result or the different result. Yeah. Yeah.
Thanks for that tour. I could probably talk for, we could all probably talk for the entire meeting about this tool, and we're excited about it. I just want to kind of jump to the vision of where we're going with this. This is one testing data set that we use. It's actually a bunch of different coffees, light roast, medium, and dark roast coffee, and we did metabolomics on it. But ooh, I need to go to a different history. This is a good thing to show to you. So it's actually very easy to jump around between different history states. They just all exist. So, you know, if you've ever had somebody come back and say, oh, we should have done this thing at the beginning of the data analysis, it's easy to go revisit it. But what we're actually starting to do in some of our own papers is show these provenance keys like in the figure legend. And the goal is that you can actually, as a core facility, prove that what you did to the data was a good and safe thing to do. So here's like a publishable, okay, maybe that's, you need to go change the labels. There's too many labels or too many rows. Oh yeah, your touchpad is weird. So here you can see we have a p-value threshold of like 0.01. Now, I'm not saying it's good or correct to just go do this, but what if you wanted to change it to 0.001? Our non-savvy core clients that don't do any programming can actually rerun that now, and you see the new plot when that's finished running. So now here's a new plot with a different p-value threshold. And what we're really starting to do, and our vision is, you could use this number, put that in the figure legend or a supplementary file, and this number proves exactly where that plot came from. So if you change any upstream steps, like for example, okay, getting a little lost here. Let's say you wanted to change, instead of a signal to noise ratio of 3 for if you found something, if you wanted to change that to 20 for, whoops, we'll do 20 here. Okay, so that will all, we're not going to watch it go, but that's going to automatically rerun through your workflow. So this is direction we're headed. We want to use some sort of, we didn't invent provenance keys, Nextflow uses them. But I think as a community, especially in this core environment, we have these competing things. We want to enable people to use our software tools, but we want to show that we were responsible when we ran it. So if we do a standard workflow, we can show that we created that history. But if they go do something like, you know, excluding a group because they don't like where that group was in the PCA plot, we don't want to stop anybody from doing what they want to do. There's valid reasons to do all sorts of things for exploratory reasons. But it's on them if they want to rerun parameters or not. So again, going way back out to a high level, I think it's very important that we adopt tools that aren't extra work. Hopefully you guys can see that this helps us do our work. We're now doing all of our data processing natively in this tool, and then we can share it. So I don't want to belabor where we're going with data reproducibility too much more in the future, but we do plan to make this all publicly available, and we're going to publish our provenance key strategy so that other people can calculate the same keys as we do without using our tool. But I think we have a very interesting opportunity as core directors to encourage adoption of these type of tools. Certainly at NYU, our plan is to start using this for core data. Now, it doesn't make sense for every core, but we're slowly rolling this out to all of the core facilities in our institution. And then in the future, maybe it'll catch on, maybe it won't, we don't know. But that's the type of motivations we're trying to balance, sharing the capability. Because I don't think, again, all the way back to the beginning of the talk, we don't think that having eLab notebook entries, having GitHub repos, and having data repositories has really achieved what we should be doing as scientists. Because I got a lot of thumbs down when I asked if running somebody else's data in a repository went well. We tried to do that. I haven't seen it go well to be honest. So I think we need to address this as a community. Like, how are we going to actually prove to the people that we do research for in our country that we're actually doing what we say we're doing? So that's where we're going. I'll leave it at that. But I want to use the rest of the time for open discussion. What tools are you using? What are the pain points? What should we be doing? What aren't we doing that we know we should be doing? So thank you for your interest in this topic. Oh, I thought there's like 10 to 15 minutes left. Isn't it till 12:15? No? Did we mess that up? Oh, no. Well, we're all available and happy to talk more later. I goofed that timing. I thought we had 15 minutes for questions. But anybody who wants to stick around, we're more than happy to chat.
We actually write because we didn't plan to show this.
Yeah. Yeah, definitely.